In this video, we're going to have a brief introduction to the idea of genetics. So genetics is the study of heredity and variation. We're going to see what these words mean as well. You can also add into this the study of genes, heredity, and variation. So we have a definition of genetics. To understand that definition, well, you have to understand a bunch of words within it. So we're going to see what heredity means, we're going to see what variation means, and then we're also going to see what genes are. So we're going to focus on the idea of characters, traits, and heritability. So you'll hear two terms. The first term is character. It's kind of like characteristic. It's just a feature of an organism. So for example, eye color is a feature, ability to roll tongue is a feature, height is a feature, flower color is a feature. You might have, let's say, the color of the pods is a feature. You might have the shape or texture of the peas is a feature. And you'll also hear the word traits. Traits is a variation of a character. So for example, the character is eye color, but the trait could be brown eyes or blue eyes. So it's a version of the character. The character could be flower color, but a trait is purple flowers or white flowers. The trait could be height, the character could be height, but the trait is the long or short for the plant. So character is the general characteristic, trait is the version of that characteristic. Believe it or not, ability to roll tongue is something that's actually determined genetically. So some of you who can't roll your tongue, or like this one here, who can't roll their tongue, well, it's because you don't have the proper genes to do it. Whereas those who can roll their tongue, they have the genes that give them the characteristics and the proteins necessary to allow them to roll their tongue. So in class, I had you guess which one of these was my mother, or is my mother. And the majority of you guessed number 1 is my mother, and some of you thought number 2 was my mother, and I think one of you thought number 3 was my mother. And there's always one who thinks number 3 is the mother because, you know, why not? Why couldn't it be? Now, most people do figure out that, again, number 1 is correctly my mother. And then some of you say number 2, and you're not too far off. This is actually my aunt, my mother's sister. And so when I ask you how you determine that, well, a lot of you point out that we have similar features, similar characters, similar traits. We have similar eye color, big noses, big lips. So we share several of these traits in common with each other. And the reason I have these traits is because my mother gave them to me when she made me. And I also got some traits from my father, and he also gave me some of the traits when he made me. So my mother essentially gave me some DNA, and so did my father, and then together that made me. So we did that activity because what we're talking about is the idea of heritable traits. Heritable traits are traits that can be passed on from one generation to the next. So from parent to offspring, and offspring just means kids. So inherited means it was passed on to somebody. Heritable trait is a trait that can be passed on. So my eye color was passed on from mother to me. Some traits are not heritable. For example, if I dyed my hair blonde, I wouldn't be able to pass on that blonde hair trait to my kids because it's not in my genes. It's something that modified about me later on. So we are studying heredity. Heredity is the transmission, passing on, of characteristics from one generation to the next. And today we know that the way you pass on characteristics from parents to offspring is the parents give some sex cells, so a sperm and an egg, and there's some DNA inside each. And the DNA from the father will be passed along through the sperm. The DNA from the mother will be passed along through the ovum or the egg. And when they combine together, they'll make you, and that's how you're determined. That's how you're formed. All that DNA has instructions on it to build you. Now, before we knew this, there were actually some other ideas about inheritance, about how inheritance worked. One of the coolest ideas I'll discuss is something called the performation theory. So in the performation theory, there were these tiny little cells called animalcules. And the idea is that let's say, for example, you were a boy, there would be a sperm, and inside that sperm there'd be a mini version of you, and eventually that mini version of you would develop into a full-grown adult. If you were a girl, inside the egg there'd be a mini version of the girl, and then that would develop into a full-blown adult. Kind of like if you watch Family Guy, when you watch the episode where Stewie drives a little sperm vehicle, that's what it reminds me of. That was called a performation theory. There were other theories out there. One of them was called the blending theory of inheritance, where people thought that if you had mother and father, let's say tall mother, short father, then you had to be somewhere in between. But as we know today, that's not the case. Sometimes you can be taller than both parents, you can be shorter than both parents. But the idea behind blending theory is that you'd be some in between of your parents all the time. But we know today that's not the case. So now we're going to talk about DNA structure in a bit more detail and relate that to chromosomes, chromatin, and genes. Those traits that are passed on from generation to generation, they're passed on because there's codes in your genetic material. Your genetic material is a big molecule called DNA. In the old days, we used to think that protein was the genetic material, but today we know it's not. Today we know it's DNA. Protein does work closely with DNA to help give you your characteristics, but it's DNA that's the main code, the main blueprint. The idea is that DNA has instructions to make proteins, and then those proteins will go and give you your characteristic. So the DNA is kind of like the blueprint, the plan, and the protein is like the material you use to build the building or whatever you're trying to build. So let's say I have in my DNA code, in my genetic code, instructions to have brown eyes. Those instructions will be read in my DNA to make brown protein, and those brown protein will go and paint my eyes a brown color. If I have instructions in my DNA to have brown hair, I'll have the DNA red, it'll make brown protein, and then that protein will paint my hair brown. It's a bit of a simplification, but that's the general idea behind it. So DNA and protein does work together, but DNA is the main control and the genetic material. So let's take a look at DNA in a bit more detail. We're going to take a look at the chemistry of DNA. So there's a bit of chemistry involved here because at the end of the day, even though we're living things, we are made up of non-living things. We're made up of these cells. We're made up of these, cells are living. We're made up of atoms and molecules that make our cells. So DNA stands for deoxyribonucleic acid, and I'm going to go over why it's called that in just a bit. But the idea is that DNA controls all of your traits. And you get your DNA from your parents. You get half of your DNA from your dad, and that's called the paternal DNA, pa, like papa. And half your DNA from your mom, that's called your maternal DNA, ma, like mama. And so the way you can look at it is, here's our cell, and inside the nucleus is where your DNA is found. And then the DNA is just unwound here, and you can see that at different parts of the DNA, there's different segments or different codes. And those codes code for different things about you. So you might have a little segment over here, a gene for your hair color. You might have a gene for how your teeth develop. You might have a gene for a certain protein in your body. So along DNA, there's all this code called genes, and those genes are read and they give you your characteristic. So let's zoom into the DNA molecule a little bit more. DNA is a very big molecule. We give it the term macromolecule. Macro just means big, so it's a big molecule. Now, the DNA is made up of smaller units called nucleotides. Whenever you have a macromolecule, it's made up of smaller units, and we give those smaller units the name mono. Mono means one, so it's a single unit. So if I was connecting, let's say, paper clips together, when I connect a bunch of paper clips together, that will be the macromolecule, but the individual paperclip is the monomer. Now, in DNA specifically, the monomer is given the name nucleotide. So you connect a bunch of nucleotides together to make DNA. That's why we sometimes call DNA polynucleotide, poly meaning many. So again, in biology, you want to try to use these key words, these prefix, the meaning of these words, to remember what these different parts mean. So again, you can see already from the nucleotide part that nucleic part refers to the nucleotide. So we have our nucleotide in our name. So this is a nucleotide over here. I zoomed in to show you what it looks like. There's 3 parts to a nucleotide. There's a sugar, there's a phosphate group, and there's something called a base, nitrogenous base. So you can bet that there's probably nitrogen in there. So here's our DNA molecule. It's usually twisted, a twisted ladder. We sometimes call it a twisted helix. If I untwist it, it looks like this over here. And so you can see here we have a nucleotide connected to another nucleotide, connected to another nucleotide, connected to another nucleotide, connected to another, connected to another. This would be considered a strand of nucleotides. That strand is connected to another strand of nucleotides. So we say DNA is double-stranded. There's 2 strands. Here's a strand of nucleotides. Here's a strand of nucleotides. What I want you to get is that if you analyze the nucleotide, just zoom into it again, we have our phosphate group, sugar, and our base over here. There are 4 different types of nucleotides in DNA, and they're determined by the base that you have, the nitrogenous base. And they're characterized by a letter. So I could have C, I could have G, I could have T, I could have A, T, C, G. And it's these letters that are actually the codes in your genes, in your DNA. So depending on the sequence of letters that you have here, you might have different proteins made in your body compared to someone who has a different sequence of letters that they have there. So the letters in the nucleotides, they're basically the code that you see when someone gives you a genetic code, a combination of those letters that determine what protein are made in your body. So here I'm just talking a little bit more about the purines and pyrimidines, or sorry, the nucleotides. So the nucleotides, like I said, there's many different varieties. There's 4 different varieties based on what type of nitrogenous base that you have. So it could be A, T, C, or G. Here I zoomed into the nitrogenous base to show you what the different nitrogenous bases look like. I want you to notice that for C, cytosine, there's only 1 ring. But for G, guanine, there's 2 rings. These are rings. For thymine, there's 1 ring. For adenine, there's 2 rings. So if you have a nitrogenous base with 2 rings, you call it a purine. If you have a nitrogenous base with 1 ring, you call it a pyrimidine. So A and G are your purines, T and C are your pyrimidines. I won't expect you to know how to draw these nitrogenous bases. You should know, though, how to draw a nucleotide, that there's a sugar there, a phosphate group here, and there is a base, and that base can be A, T, C, or G. I want you to notice that the sugar has a name. The name is deoxyribose. Now you know where the deoxyribo comes from in DNA, deoxyribonucleic acid. Deoxyribose is for the sugar, nucleotide, and that explains our name. So again, I do need you to know the general structure of a nucleotide. Over here we have a phosphate group, we have our sugar, and we have our base. Now you might notice that this is a purine. So you would know, because you would know that this is a purine because there's 2 rings, and so that means it's either A or G. To be more specific, you'd have to know the structure in more detail, and there's differences that you could look up later on. Over here, we know this is a pyrimidine because there's 1 ring. So that would be C or T. So I could ask you if the base you have is a CT or AG, and you can figure that out using your purine versus your pyrimidine knowledge. So now we'll go into the chemistry of the DNA molecule in a little bit more detail. Like I said, the DNA is often called a double helix. Helix refers to the fact that it's a twisted molecule. It twists around like a little twisted staircase. And double means that there's 2 strands of nucleotides. So we have a strand of nucleotides over here and a strand of nucleotides over here. The rungs, like the step over here, the steps of the ladder, those are your bases. Those are your nitrogenous bases. So this is a rung of the ladder, a nitrogenous base, nitrogenous bases. nitrogenous bases. The posts, the sides of the DNA, that's just sugar and phosphate. So we call it the sugar-phosphate backbone. The two strands, they're held together by hydrogen bonding. So you can see A and T are held together by 2 hydrogen bonds, C and G always by 3 hydrogen bonds based on the nature of their chemicals in them. So A and T always 2, C and G always 3 hydrogen bonds. So in class we were trying to discuss which ones would be easier to break, and you guys were able to figure out that, yeah, A and T are easier to break because there's only 2 hydrogen bonds compared to 3 hydrogen bonds. In the sugar-phosphate backbone, it's all strong covalent bonds. It's not hydrogen bonding. It's really only between the bases. Now you'll notice that bases always occur in pairs. So here G is connected to C. Here C is connected to G. A is connected to T. So we have our base pairs. They're called base pairs. And what you may have noticed is that whenever you have T, it's always connected to an A. Whenever you have G, it's always connected to a C. A to a T. So it's always A, T, C, and G. So for example, here I have my single strand of DNA. You could actually figure out the other strand by knowing that A always goes with T and C always goes with G. This blue part here is my sugar-phosphate backbone. Here are my bases. So if I have C over here, I should have G over here, and a sugar-phosphate backbone, of course. I should have A here, C here, G here, A here, T here, A here, G here, C here. So when we're trying to figure out a genetic code, if I know one strand, I can figure out the other strand because they always have the ATCG. That's actually called complementary base pairing. So as I mentioned, complementary base pairing is the fact that A always pairs with T and C always pairs with G. When I say DNA sequence, that means the order of nucleotides in a strand. The order of nucleotides in a strand. So I might have T, C, A, T, G, C. That's a DNA sequence. So in this picture, if I were to ask you for a sequence, if I were to focus on, let's say, this strand over here, it would have the sequence A, G, T, A, C, G. The numbers represent the direction that you're reading it from. So if you're reading it from the phosphate end, that's given a number 5'. And what you don't see here is at the end here, there's actually a hydroxide group where there's no phosphate. That would be the 3' end. So this is going from 5' to 3', and I just indicate that for direction purposes. Over here, since there's no phosphate, you know it's a 3' end. And over here's a 5' end, so this strand goes in the other direction. So you have 3', T, C, A, T, G, C, 5'. So you'll notice that they go in opposite directions. 5' to 3' over here, 3' to 5' for this strand over here. So again, if you know the direction and bases on one strand, you can figure out the direction and bases on the other strand. So if I have my 3' end over here, then my 5' end should be here, because they're opposites, and my 3' end should be here. So my C, there'd be a G paired with it, an A with my T, C, G, A, T, A, G, and C, just like that. So I'm just going to show you how to build a DNA molecule. So if I were to, I'd first start off by building a strand. So you can see in this animation that I'm connecting the nucleotides together. The nucleotides are connected together by covalent bonds. And those covalent bonds are given a special name. It's called phosphodiester bonds. I would then build another strand with some strong phosphodiester bonds. And then I'd connect the two strands using some hydrogen bonds. So it's much easier to break the strands, to separate the strands from each other, than it is to break the individual strands. Because the individual strands, they have phosphate bonds, covalent bonds, or I should say phosphodiester bonds, that are very, very strong. Whereas between the strands, you just have hydrogen bonds, and they're pretty weak. I mean, they're strong together, but they're weak compared to the covalent bonds that exist between the nucleotides. So just a little summary here. Phosphodiester bonds are the strong covalent bonds that exist between the nucleotides in a strand. They're within one strand. The hydrogen bonds are the weaker ones that exist between the bases of the two different strands. So 3 for CG, 2 for AT all the time. And of course our lame joke, the James Bond. This is just a little practice slide for you to attempt. You have the nucleotide sequence, the DNA sequence of one strand, and you have to figure out the DNA sequence of the second strand using the complementary base pairing rules. And then you're also told to draw the correct number of hydrogen bonds between each base pair. Remember, 2 for AT, 3 for CG. So like I said earlier, the DNA is also called a double helix. It means two strands, but it's also a twisted ladder. That's what the helix part means. So we can see our two strands over here. This is one strand, this is the other strand, and it's twisted because of the nature of the chemicals that make up the DNA molecule. So now let's take a look at what a gene is. A gene is a specific sequence of nucleotides. So you know what nucleotides are now. They have the letters to represent what they are. It's a specific order of them that codes for something, that codes for a particular trait or protein or whatever be the case. So in your DNA, this sequence of letters will code for your traits, for the traits of your characteristics. And you get this DNA from sperm and egg of your mom and dad. And basically in your cell you have this machinery, these parts that read those letters and then know what to do with it. They'll say, oh, this is a letter for a brown protein for brown eyes. This is a letter saying that there's no brown eyes, that there's blue eyes instead. And that's what a gene is. It's a sequence of nucleotides that codes for something specifically. Now the people who discovered DNA, there's actually quite a few involved. There's two male scientists, James Watson and Francis Crick, and then one female scientist, Rosalind Franklin. Now, I'm not going to speak too much about them, but what I want you to know is that science is actually oftentimes full of drama. I mean, people are racing to make discoveries. Rosalind actually did a great deal of research to find out the structure of the DNA molecule, and these two, they used her work and came up with the structure without giving Rosalind any credit. Unfortunately, she didn't get a Nobel Prize for her discoveries. She was given very little credit for it at the time. Women in science were often not recognized for all the great work that they did back in the day. So now that's something that we're trying to change. Women make a lot of discoveries in science, and when it's time for them to get recognition, they get it now. There's still a lot of work in the way of that, but it's very, very important that, you know, if you're a female, that you have an interest in science, that you pursue it, because there's lots of promise in the field, and it's something that you should continue. So, DNA. In your cells, in each cell that you have, there's about 6 feet of DNA, 2 meters of DNA. That's 2 meter sticks. That's a lot of DNA. And it has to be packed into this very, very small nucleus. It's 0.000006 meters in size. So it's a very, very small nucleus. We're somehow packing 6 feet of DNA, 2 meters, into this over here. So you can think of it kind of like fitting 20,000 meters of string into a tennis ball. So how do we do that? Well, we wrap the DNA around stuff. Kind of like if you were putting away a plug or some kind of cord, extension cord, you'd wrap it up around your arm or around something to keep it compact. And that's exactly how your how your body does it. There are special proteins in your body called histone proteins, and you wrap the DNA around the histone protein, and that way it can fit into your super, super, super tiny cell, and even more impressive, the super, super tiny nucleus of the tiny cell. So here we have a picture of the DNA being wrapped around histones. So you can see our DNA molecule over here is wrapped around this cluster of protein called the histone proteins. And essentially you keep wrapping it, keep wrapping it, keep wrapping it, and eventually your DNA will look like this. It'll look like chromatin when it's not fully packed. And if you make it a bit tighter, it'll look kind of like this. And that's called chromosome. So when you heard those words chromatin and chromosome before, that just basically means your DNA wrapped around protein, but at different tightness. So for chromatin, it's loosely wrapped. For chromosome, it's tightly wrapped. It's more compact. But it's still DNA wrapped around the same protein called the histone protein. When you wrap DNA around one of these little histone proteins clusters, it's now called a nucleosome, and then basically a whole bunch of nucleosomes get wrapped together, and that eventually becomes chromatin and chromosome. So let's quickly talk about how DNA looks like inside the cell. So DNA can look different depending on the phase of the cell cycle that you're in. Chromatin is when DNA is loosely wrapped around protein. It looks like spaghetti. Loosely wrapped around protein. You usually find chromatin during interphase, when the cell's not dividing, when you're not really moving the DNA around too much. So when the cell's not dividing, you're going to find DNA in the form of chromatin, and like I said, it looks like spaghetti. When you look at a cell under the microscope, if there's chromatin, it looks like a nice smooth nucleus, because the chromatin is very thin, chromatin thin. So here's our chromatin in a cartoon, and here's our chromatin under a microscope. It's the blue stuff, and it's nice and smooth because, again, the chromatin is very thin. You can also find DNA in the form of chromosomes. When you find DNA in the form of chromosomes, they can look like two things. It can look like this, or it can look like this. But the idea is that DNA in the form of chromosomes is just DNA wrapped tightly around the histone proteins. And you usually see chromosomes when the cell is dividing. So usually during mitosis, meiosis, that's when you see chromosomes. Under the microscope, the chromosomes look like worms because they're a lot thicker than chromatin, so it's not as smooth. You can actually visibly see the thick chromosome structures that are currently in the cell. So here you can see prophase. The chromosomes have started to condense. The DNA has started to condense by wrapping itself more tightly around the chromosomes, and you can see those little worm structures there because the chromosomes are a lot thicker than the chromatin. And the reason that usually we want our DNA to go from chromatin to chromosome is because during mitosis and meiosis, you have to move the DNA around. And it would be a bit difficult to move your DNA around if it was in the form of chromatin. It's all tangled up. It's organized but tangled up. So it's much easier to compact your DNA into chromosome form so that it's a bit easier to move. Kind of like if you were moving out of a home, you would take all your stuff, pack it in a box. You compact it into one small area so that you don't forget anything. It's nice and easy to move around. So where do genes fit into all this with the chromosomes and DNA? Well, here, hopefully now it all kind of fits together. Here we have a chromosome. The chromosome is basically just your DNA wrapped around protein. And so here, this little band here, that represents a sequence of nucleotides that codes for something important. It represents a gene. And so this gene on the DNA that's found on this chromosome might be a gene for eye color, and it codes for brown. Over here we have another gene on some other area on a chromosome, and that codes for hair color, and it just happens to be hazel hair. So your chromosomes contain your DNA, and your DNA contains your genes, and those genes are the instructions for what you should look like. The location of a gene on a chromosome is called the locus, a locus. So we might say the locus for eye color, the locus for hair color. Plural of locus is loci. So there's some gene loci or a gene locus, singular. Now humans have about 19,000 to 20,000 genes. And all those genes are found on their chromosomes. Specifically, they're found on 46 chromosomes. So in your cells, in your body cells, there are 46 chromosomes. And those 46 chromosomes have all your 19,000 to 20,000 genes. So you can see here we have a baby. If you zoom in, you can see its cell, and you can see its nucleus. If you zoom in the nucleus, you'd see its chromosomes, or chromatin. If you zoom into that, you would see, well, the DNA. Eventually you keep zooming in, you'd see the proteins, but keep zooming in beyond the proteins, and you'd see that it's actually just DNA there, wrapped around the proteins. So we're going to finish off by talking about the difference between somatic cells, gametes, and we're going to do a brief overview of the main division processes that exist in organisms, and we'll take a look at the different types of reproduction that exist as well. So one type of cell in your body is called a somatic cell. Those are your body cells. Those are the cells that are not sex cells. They're not sperm, they're not egg. They're things like skin cells, heart cells, liver cells. And each somatic cell in your body, in humans, have 46 DNA molecules, and you could also say 46 chromosomes. The other type of cell is called a gamete, and that's a sex cell. It's used for reproduction. The sex cells can be sperm or the egg, also called the ovum, more specifically. Gametes don't have 46 DNA molecules. They have half the amount, because if you think about it, when parents make you, they don't want to give you double the amount of chromosomes that you should have. Humans should have 46 chromosomes. So if your dad gives you DNA or chromosomes, it has to be 23, and your mom has to give you 23. So when you add them together, it's 46. So when sperm and egg are being made, there's actually a special type of division to get half the DNA in each. I want you to get that not all organisms will have 46 chromosomes in their cells. Dogs, for example, have 78 chromosomes in their somatic cells, but that means 39 chromosomes in their sperm and egg. A cat has 38 chromosomes, and that means 19 chromosomes in the sperm and egg. So different species, different organisms will have different amounts of chromosomes in them, but the idea behind somatic and gametes still applies in terms of the number of chromosomes that they would have. So another important term you need to know is the term diploid and also the term haploid. So somatic cells, they are called diploid, and we represent diploid with the letter 2N. And the reason they're called diploid is because they have two sets of chromosomes. One set came from dad, so we call that N. That's the one set from dad, 23. And the other set came from mom. We also call that N, and there's 23 in there. And so when you put them together, that makes your zygote, or your somatic cell, that's 2N, is equal to 46. So if you have two sets of chromosomes, you're considered a diploid organism, whereas human gametes are haploid, for example. So haploid is represented by the letter N, and they're haploid because they only have one set. So if you look at these pictures here, the red, there's two of the red. One probably came from mom, one from dad. Two of the purple, two of the yellow. So because there's two of each, we know there's two sets of chromosomes, whereas here there's only one of each. So that we know that that's haploid. So here we can see our human cell, and you'll notice that there's two of each of these chromosomes here. So there's two sets of chromosomes, whereas in the sperm there's only one of each, so it's haploid. So diploid, two sets, haploid, one set. So if I was an organism that had 14 chromosomes and I was diploid, I'd say 2N is equal to 14. In my gametes, I'd have N is equal to 7. This would be diploid, this would be haploid. So you should be able to answer which, you should be able to do this question now to figure out which cell's haploid, which cell's diploid. So if you take a look carefully, Over here we have two of these cells, two of these chromosomes, two of these chromosomes, two of these chromosomes, two of these chromosomes. So we have one set that probably came from dad, one set from mom. So because we have these chromosomes in pairs here, we know that this is diploid. There's two of each type, two of each length, let's just say. Whereas here, there's only one of this one, one of this one, one of this one, one of this one. They're not paired up. There's only one set. So this would be haploid. The cell on the right's haploid. And the cell on the left is considered diploid. And my explanation was sufficient to explain how I knew that over here. Now, believe it or not, you could actually have more than diploid. There are organisms out there that can be polyploids. Poly meaning many. So more than two sets, three or more. So here we have our haploid, only one of each chromosome. Here we have our diploid, two of each type of chromosome. But notice here we have a triploid, three of each type. Here we have a tetraploid, four of each type. Believe it or not, strawberries are octoploids. Yes, that's right, 8 of each type. Usually polyploidy is more common in plants than in animals. There are some animals that are polyploid, so for example, there's some rodents that are tetraploids, and they just have bigger sperm than the diploid version. They also have a bigger tail. Usually you have larger sperm if you have higher ploidy, just because of the larger amount of chromosomes or DNA that's found within it. So let's now go take a look at how you became you. We're going to see all these different processes of division. So the idea was that your father had some sperm, and inside that sperm there were 23 DNA molecules or 23 chromosomes. Your mother had an egg, and in that egg there were 23 DNA molecules or 23 chromosomes. The sperm and the egg combined. So when you combine sperm and egg, it's called fertilization, and you get a fertilized egg called a zygote. This here was haploid. It was N is equal to 23. One set. This here was haploid. It was N is equal to 23. One set. This is now diploid. Two sets. 2N is equal to 46. And so through the process of mitosis, that cell just divided and divided and divided and made a larger organism. Mitosis just copied that cell over and over and over again to make a larger organism. Now, that's one of the types of division that you're going to see, mitosis, to make a multicellular organism. But keep in mind, something had to make these gametes. Another type of division that we'll explore is meiosis. That's the division that makes gametes. Mitosis makes cells with 46 chromosomes. It doesn't change the number of chromosomes, it preserves it, keeps it the same. Meiosis makes cells with half the number of chromosomes, so that when they combine, they can have the correct number to begin with. So here we have the fertilization event. This is a bit disturbing, I apologize. This is how it was made. Sperm, fertilized egg, zygote, and then mitosis happens, and you get an adult. So there's two types of reproduction that I want to discuss, and they have their advantages and disadvantages. So asexual reproduction is the first part, asexual. Usually when you add a to a word, a means without, so without sex reproduction, no sex involved. Asexual reproduction is when there's one parent that gives rise to offspring, and the offspring are genetically identical to each other, meaning they're clones. So there is an advantage to this. First of all, there's no need to find a mate. You don't have to do anything special for your mating. There's no mating behaviors or dances. There's no need for any specialized organ, and it's pretty quick. It's really, really fast. There's not much risk. It's pretty certain. You'll engage in the activity by splitting, and you make your babies, and that's it. The disadvantage, though, is that you made clones. They're all the same. They're all genetically identical. There's no variation, and that's bad. You need to have variation to have a healthy population. If everyone was the same, everyone would have the same weaknesses. So if one person was sick and died from a disease, everyone else would die from that disease as well because they all have the same immune response. But if you have diversity, then you're more likely to keep that population surviving because some might have stronger immunities than others. So asexual reproduction, no sex, one parent gives rise to offspring. The offspring are genetically identical. There are advantages. It's not risky. It doesn't take up much energy. There's no specialized organs needed, and it's pretty quick. But the disadvantage is everything's the same. Everything's kind of boring. So there are many plants that will do asexual reproduction. I want you to take a look at the different examples of asexual reproduction in your textbook. That would be good to maybe write down and add on to your notes. Here we see a bacterial cell doing some asexual reproduction. This is called binary fission, where the cell essentially splits into two identical daughter cells, and it happens really rapidly. Mitosis is another example of asexual reproduction. There's only one parent involved. Now we'll take a look at the other type of reproduction called sexual reproduction, where there is sex involved. So sexual reproduction occurs when you fuse sex cells from different parents. So there's a few individuals involved. Okay, two individuals involved, they fuse sex cells, they fuse gametes. Now, usually those gametes come from two different parents, but sometimes they can be part of the same parent, but they still combine gametes in different ways. But when we discuss sexual reproduction, we're going to take a look at the reproduction from sperm and egg. Now, there is a disadvantage to sexual reproduction. First of all, it requires specialized organs, and it can be energetically costly. You have to do mating behaviors. You have to go look for a mate. You can be kind of risky too. For example, if you look at this peacock, he has very, very bright colors to attract a mate, to attract a female to have sex with and reproduce with. And yes, it will do the trick. The males love it. The females love this. But what else loves this? What else can see this? That's right. Predators can see this. So it's a bit risky too. And sometimes you might have a combination of genes that makes the offspring sick, so they can't survive. So you might be wondering, well, why not just do asexual reproduction? Why is there sexual reproduction? Why not just split into two and then that's it? It seems a bit easier, even though there's no diversity, it seems a lot easier and a lot better. But there's something that keeps sexual reproduction from disappearing, something that keeps sexual reproduction advantageous. And the main advantage is that sexual reproduction results in offspring that are variable in diversity. And that's what allows organisms to survive in environments that are rapidly changing. If everything was the same and there was a change in the environment and that thing killed one thing, everything else would die. But if everything was different, if there were differences and there was a change in the environment, the population could at least survive and reproduce and pass on its genes. So generally, sexual reproduction, shouldn't say no sex, it should say sex over here. Generally, sexual reproduction is more advantageous, especially in those changing environments where diversity can allow the population to survive. Asexual could be advantageous in an environment that's not changing very much, as long as the organisms have adapted to it to begin with. So generally we say it this way: sex is costly to the individual, but beneficial to the species. And sex is especially beneficial in the environment where it's constantly changing. So yes, sex requires energy. Yes, it might require mating behaviors, but it results in variation, which allows species to survive when their environments are changing. So this is a summary of the different types of reproduction that we saw. We saw asexual reproduction and sexual reproduction. You should know what the differences are between the two, what the main definition is, and also what the result is, and also what are the advantages and disadvantages. So you can take a look at the advantages of sexual reproduction over here, disadvantages over here, advantages of asexual reproduction over here, and disadvantages over here. So do take a look at the difference between the two and understand the advantage and disadvantage between them. The reason we discuss these types of reproduction is because there is a strong tie to evolution here, but also because mitosis is a type of asexual reproduction. And when we discuss meiosis, we're producing gametes so that we can engage in sexual reproduction. So it is connected in those two different ways. This is your homework that you can practice for this PowerPoint. There's a reading, page 138 to 143, along with the questions that you see over here. And there's also some slide questions that you can work on once you've gone through this PowerPoint and this video.